All right. Hello, everyone, and welcome to today's keynote presentation titled Widespread Unappreciated Alpha-Synuclein Aggregates in CNS, a Cause of Symptoms, Caution, and Optimism in Parkinson's Disease and Lewy Body Dementia. This webinar is a part of the 12th annual event in the Neuroscience Virtual Event Series. I am Sydney McNeil of LabRoots, and I will be your moderator for today's event. Today's web seminar is presented by LabRoots. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits image located on the right of your screen and follow the process to obtain your credits. Now let's get started. I now present today's speaker, Professor Paul Henning Jensen, Aarhus University, Department of Biomedicine, Director, Danish Research Institute of Translational Neuroscience, Nordic EMBL Partnership for Molecular Medicine. For a complete biography on our speaker, please find it on the left of your screen. Professor Paul, you may now begin your presentation.
Thank you very much, Sydney. And just first, my name is Paul Henning Jensen. I'm basically an MD, but I'm a basic scientist studying a protein called synuclein and its involvement in neurodegeneration. I'd like to thank the organizers for giving me this opportunity to present the data. Just for you to know, I have no cursor, so I'll try to help you navigate around in the slides as good as I can. But basically, I mean, what I would like to talk about is what I call widespread unappreciated synuclein aggregates in the brain. So this protein is important. We believe we know where it is, but I would argue we don't really know that. And by saying that, I would say this could be a cause of what do we say here? Yeah, then we need to discuss what do I mean by appreciated versus unappreciated. I'll get into that. And then I would also, I need to get this moving. Then I'll also say that I'll try to explain why I think this could be relevant for symptomatology. It should also be a cautionary note when we try to study these diseases, because we might actually miss quite a lot if we don't pay attention to this. And I think it's also a reason for optimism, but I'll get to that a little later. So the agenda for today would be some kind of, because this is a broader audience, so I'll give some Parkinson's disease basics. I'll talk about alpha-synuclein's role in Parkinson's disease. And then I'll talk a bit about how we actually perceive synuclein aggregates in brain. I mean, what is it we see? And how can we see more? Then I'll show some surprising new findings that has come within the recent years, where we have used some let's say, newer techniques. They're not novel now. And finally, whether it could have consequences for moving forward. And then just before we get into the show, I'd like to thank, I mean, this I present is basically data that has, accumulated in my group for many years. So I won't mention all the names here. I'll say I'll show unpublished data that has been generated by my former PhD student, Nana Møller Jensen, in collaboration with Yu Hong Fu and Glenda Halliday in Sydney, and also Silvio Rizzoli in Göttingen. I'd also like to thank the foundations that have supported our group for many, many years. And then I'll just tell that I have no commercial interest to disclose for what I'm presenting here. So let's get to it. Parkinson's disease, let's say, if we take it from the perspective of the patient or the relatives, it's a progressive disorder. It don't get better. Typically, you realize you have the disease when you have the classical motoric symptoms. That is this bradykinesia, slowness of movement. You have this rigidity, stiffness of your arms and legs, and you can have a resting tremor and some other depicted to the figure to the left. These symptoms typically come when you have lost about 50% of your dopamine-producing neurons in an area called substantia nigra that's highlighted in red. These neurons project their terminals up to the striatum, and that's where they deliver dopamine, and that's important for our normal motor coordination. However, when you get the diagnosis, you typically will be treated with L-dopa or a dopamine agonist, and you will feel this works very, very well for you. But you might also have other symptoms, and that's what we call the non-motor symptoms, and they can have different flavors. What I've highlighted in red is what we could call cognitive or psychiatric disorder symptoms like anxiety, depression, cognitive problems. You could also have symptoms that arise from your autonomic nervous system. Could be incontinence, orthostatic hypotension, sweating, constipation. And then there's a series of other symptoms that can be pain, sleep problems, dysphagia, difficulty to swallow food. These symptoms are not well treated with our current therapy. So in a way, we can say we are not good in treating our patients, but we are fairly good in managing their motor symptoms in the beginning of the disease. So just to make it clear here, so Parkinson's disease is a progressive disease, and it affects both the central and the peripheral nervous system. Now, before year 2000, one would say to make the diagnosis of Parkinson's disease, this is from a very nice paper, it said that the pathological diagnosis is fairly easy. You just need to see that there is a substantia nigra degeneration, and you need to see Lewy bodies. And as it says, this simple matter is what the neuropathologists need to make or refute the diagnosis of Parkinson's disease. This being said, it's not just substantia nigra that's degenerating in this early stage. Also, locus coeruleus, a little lower in the brainstem, is also losing as many neurons as the substantia nigra. Now, then, if we say this Lewy body, you need to make the diagnosis. What is that? I mean, for many, many years, it was something that was easy to stain with just some dyes. Recent data is just shown here. One of them is where you use cryo-electron tomography. You can actually show that a Lewy body is a large multi-organelle inclusion in the neuron. It contains lysosomes, mitochondria, synuclein, ER membranes, and vesicles, and so on. So this is not just a simple thing. It's something that the cells has built. So I would say the Lewy body is the appreciated pathology of Parkinson's disease, and it contains alpha-synuclein. Then, studying Lewy bodies, Heiko Braak in 2003 was able to come up with this hypothesis where he said, yes, but you actually have Lewy bodies in your brainstem lower down than substantia nigra before you get your diagnosis. Then it spreads up into the brainstem. When it reaches substantia nigra, you'll get your diagnosis. And then it spreads further on in the nervous system, and then you get, the more advanced symptoms like dementia that often comes after many years of the disease. So this actually showed that Parkinson's disease is more than just substantia nigra or pars compacta degeneration. And it also created this concept of spreading of the disease. Actually, it could come from the gut, because the first area to see the Lewy bodies within this, the motor nucleus of the vagus that actually innervates the gut. So that was novel here. Then recently, it has been said, yeah, but maybe there's more to it. We could have this, what they proposed here was the body first, where the disease starts in the gut, comes up into your central nervous system, either via the vagus nerve, but it could also be via the sympathetic ganglia. Or, and that's the novel thing in this hypothesis, it could actually initiate in the brain and then basically spread the other way around. And by having this, you could say these different origins of the disease will cause a different, let's say, sequence of symptoms to arise. And this, I would say, this is not, agreed across the field, but these are, the hypotheses, and I mean, I kind of like them, but, this can be discussed. Now, then, what is alpha-synuclein? It's a small presynaptic protein that is highly enriched in the nerve, in the presynapse that's depicted to the right here with a red dye. So you have the nerve cell, you have the long axon, and then in the end of that axon, you have the terminals. That's where alpha-synuclein is living. What I've shown to the left is basically just a stain of a piece of brain with an antibody, asyn 211, you can buy it at Biochem. But I mean, it's one on one main. But the point is, this is a nice picture because it shows these small dots, that's pre-synapses. And it also shows that the cell body is basically more or less devoid of synuclein because there's this very efficient transport to the terminals in the normal cell. Now, then one could say, is synuclein important for alpha-synuclein, for Parkinson's disease? Well, I mean, the reason we believe it is based on genetic and also pathological data. But if we go to the left, we see the genetics part. It has been shown many times in solid that if you have mutations in alpha-synuclein, if you have insertions, this was recently shown actually if you insert a stretch of amino acids in your synuclein, or if you have multiplications of the normal gene, so you code or express more of the normal synuclein, then you will get what we call an autosomal dominant Parkinson's disease and Lewy body dementia. But it has also been shown that variations in the synuclein locus, it's called this SNCA, is actually a risk factor for idiopathic Parkinson's disease. So this seems to be affecting, the larger population because these familial cases are rare. If you look at the pathology and look to the right now, then you'll see this kind of cartoon in the top where to the left you see these small strings. That is the native synuclein. And then to the right you'll see these filamentous forms. That's what we believe are present in Lewy bodies to some extent. And you will also see some immunohistochemical stainings where the brown is the stain with an antibody to synuclein. So you can see suddenly synuclein is actually present in the cell body and not just in the terminal. And below you can see this small gray picture. That's an electron micrograph where the black dots are antibodies that bind synuclein and show that this filament from a brain affected by Parkinson's disease actually contains alpha-synuclein. Then here, how do we detect synuclein aggregates? And that means the synuclein pathology. I mean, originally it was a stain with eosine, for example. So you had this, what you'll see in this picture, this pinkish round thing in top of the brain cell you see there. This is just the stain with a dye. Later on, one could use immunohistochemistry and use antibodies to ubiquitin. You will see the same structure being lighted up. After 1997, we discovered that synuclein was involved in Parkinson's disease. We started to use antibodies towards alpha-synuclein. And then you could see what is depicted here. Again, these brown spots that are the Lewy bodies. And we also saw this elongated structure you see in the top left, which are basically what we call Lewy-neurites. That's axons that are swollen and where these synuclein aggregates are also accumulated in these inclusions. Then later on, it was discovered that there was a modification on serine 129 that's phosphorylated. And if you made an antibody to this specific modification, then you get what you see to the right. To the left, you'll see an antibody to synuclein. You see the Lewy bodies, but you see a lot of brown blur around it. If you just label the phosphorylated synuclein, then you get a much more clear picture where you see the Lewy bodies, you see the elongated Lewy-neurites, but you don't see this blur of normal synuclein. This is basically typically what people use today to detect abnormal synuclein with immunohistochemistry. And then within the last, let's say, 5-6 years, it has been more normal also to use antibodies that selectively, or at least preferentially, binds to aggregated forms of synuclein. And that's what you depict here with an aggregate selective epitope. So again, what I showed you here is the appreciated neuronal alpha-synuclein pathology. You can also see alpha-synuclein pathology in glial cells. And for example, in the disease called multiple system atrophy or MSA, there, and that's the surprise in this disease, there you see the inclusions in the oligodendrocytes, and they are called glial cytoplasmic inclusions. This is a very severe Parkinsonistic case. You can also, in multiple system atrophy, if you look carefully, and some do, but it's not typically what people speak about, then you can also appreciate that there are actually some neurons that express or have synuclein present in the nucleus or in the cell body. And you can also see it in the lower right corner, actually in neurites. That's probably axons. I would argue these are less appreciated, and we don't really know the relation between what's present, the large abundance in oligodendrocytes, and what's present in neurons. But what we know with this disease is that there's actually quite a severe loss of neurons and also quite severe neuroinflammation. Then in Parkinson's disease, in short PD here, it was shown by Heiko Brack first that astrocytes also express synuclein, which is abnormal. What it does, we don't really know. It may, trigger some kind of neuroinflammation. Recently, it was shown that the synuclein in astrocytes is probably truncated in the N and C terminal part and is also modified on tyrosine 39. This was based on use of specific antibodies. Now, then we shift gear. And then so now let's talk about the aggregates. Again, you see this aggregation of the native to the filaments. There's something in between. Now, these filaments, they are formed of two protofilaments. That's the two to the left, the blue ones. And they can associate and make a proper fibril. What we realized the recent years is that the folding of the protofilament, the three-dimensional organization of these amyloid structures can actually differ. And also the way the two protofilaments associate together and make the filaments. So there we say they can have different strains. You have basically different folding strains. This was first shown in 2013 from Ronald Melke's lab, where he did simple experiments by simply letting synuclein aggregate, and then he changed the buffer conditions, ions and so on. And what he could show was that he could make two very different strains simply by changing the buffer conditions. And this was depicted here using an antibody that recognized specific epitope that was only on one filament and not the other. So that was, an in vitro strain. To show that these strains actually have, you could say, does it matter? And then what we did in our lab, and it has been done in other labs too, was simply to make two different strains. Let's say just for simplicity, a blue strain and a red strain. They differed. Then we injected them into a mice model that's overexpressing alpha-synuclein, and then we could see, as expected, that the mice started to die after about 70 days. And what we could show here, what you see in panel A, is that the red strain, they die faster. You could also see in panel B, they develop symptoms faster. The symptoms here are hind limb clasping. And in panel C, you could say, okay, from the time of symptoms to the time of death, it was way faster with the red strain than the blue strain. So again, the folding of the synuclein, it's the same monomer that built them, actually changes its impact in the nervous system. The recent years, based on cryo-electron microscopy, and it has been, we have been able to study filaments that is extracted from human brains. And it has to a large extent been dominated by Shear's group in UK. Here's a review that I referred to. But what they've shown is that the filaments from Parkinson's disease, dementia with Lewy bodies, and Parkinson's disease dementia probably look alike. They could also show that MSA, they are different. And within one single brain, you can actually have different strains. And finally, there was this juvenile onset synucleinopathy. It was recently shown where they had this insertion in synuclein. There you actually had a different fold as well. So it's just to say that what we make in the lab is actually something that reflects something that goes on in the brain, even though we might not achieve the folding that is in the brain. So I would argue again, now we get closer to this unappreciated level of disease phenotyping, the different strains, because this is difficult to appreciate. There's a large effort being done where people try to make strain-specific probes. For example, this paper here where they use some small molecules that intercalate and give fluorescent signals. But also, this was a very small paper from our group where we could also show maybe we don't capture the whole, let's say, diversity of strains, because what we could show was that the detergent usually used to extract these filaments actually destroy some of them more than the others. Yes, I think that was it. Now, let's then move to, the real unappreciated aggregate pathology. And I don't know whether my picture is really illustrated or not, but let's see. Below right, you see this native unfolded form, these small sausages. I would believe they're probably what is present in the nerve terminals. Then when they really aggregate and make the amyloid fibrils, then they probably accumulate in the Lewy inclusions. That's shown to the right with the immunohistochemistry. And then in between, we might have some, let's say, smaller aggregates. It could be smaller filaments. It could be some very different structures. We don't really know. How do we detect them if they are not abundant? If they are smaller, then they might also have an easier way of moving around within the cells. They might have an easier task of interacting with, let's say, organelles or other molecules. It may be easier for them to move between cells. I don't know, but they could actually be more bad to have than a rock-solid filament. But how do we detect them? Immunohistochemistry that is used for the two pictures you see here, they basically detect bulk accumulation of an antigen. So if we have something that's not abundant, then immunohistochemistry is not really the way to go. And then one could ask, did Heiko Braak, when he made this, depicted this spreading, did he get the full picture, or could there actually be a spread of aggregates way before and in other areas than what he saw when he stained for the Lewy bodies? Now, how do we go for that? In 2015, there was this paper with the first author, Roberts, where he said, okay, could we try to ask this question in a different way? So if I have an antibody to synuclein, and I have the same monoclonal antibody to synuclein, and they bind, and they are close together, and this can be detected with a technique called proximity ligation assay, then we probably have a small aggregate. At least we have a dimer. So then he used this method. Very simple. This tool has been used to study interactions between different molecules. He just asked, is one synuclein close to the next? Then it's probably an aggregate or a dimer or a trimer. And it turned out to be fairly productive because it was used by some groups, here's some Japanese very strong papers that came up with these surprising findings. Just the title says there's a discrepancy between the distribution of alpha-synuclein oligomers, that's what he detected with the proximity ligation assay, or in short PLA, and then the Lewy bodies. And what you see on the top right is the occipital cortex that is typically considered devoid of Lewy bodies. So if you were, let's say, Heiko Braak, or I don't know whether, but he would probably say this is not affected in Parkinson's disease. But when they use this proximity ligation assay, they could actually see a lot of let's say, PLA positive aggregates. So there might be something going on there, but we could not appreciate it. The same group went on, and then they studied multiple system atrophy. As I told you before, this is a disease where we kind of said, yeah, in multiple system atrophy, we have the pathology in the oligodendroglial cells. What they showed was that there's a wide distribution of synuclein oligomers also in neurons. And that's what I'll show you here. So these are basically, let's say, more or less parallel sections. You see here a few to the right, discrete glial cytoplasmic inclusions. But to the left, you can see the PLA signal is much more widespread in the tissue. So it seems as if there's a much more widespread burden of aggregates that can affect the tissue's functionality. So is this relevant for disease modeling? I would argue it is because, typically when we try to model, for example, multiple system atrophy, more of our models is based on the expression of synuclein in oligodendrocytes, but that might not be the whole story. But of course, it's an attempt and it works for something, but it's just, there's a layer we don't get. Then Nana Møller Jensen in my group, she kind of asked, the question, how early does these PLA positive aggregates develop in Parkinson's disease? She used a different antibody that they used in the former papers. This is an aggregate-specific antibody rather than just a synuclein antibody. So here she says, if there's an aggregate-specific epitope and there's an aggregate-specific epitope next to it, then we probably have an aggregate. I mean, this was done in collaboration with Yu-Hong Fu and Glenda Halliday from Sydney because they provided all the tissue and did a lot of the work for us as well. And then we said, okay, let's try and get a piece of tissue that's not affected early in Parkinson's disease, but late. And what you see is from the motor cortex, Then you'll see we stain with the antibody using immunohistochemistry. So to the control, you see this dotted stain, that's nerve terminals where you have some staining there. And then in the late, you'll see a Lewy body that's depicted red. When we quantify it, you can see that there's a large increase in the late states, as expected. Now, then when we made the PLA, then we basically saw this picture here. No signal in the controls, but in the early cases where there were no Lewy bodies, we actually had the tissue saturated with the PLA signal. So there has actually been a spread of something abnormal to the tissue that was unappreciated. And then when she kind of quantified and said, okay, how many neurons are affected? Then if you go to the lower part of this table, then you can see this is percent of neurons that contains Lewy bodies. So in the late stage, 4% of the neurons are carrying a Lewy body. That's what you would expect to see. But when we say, okay, how many of the neurons are actually positive for this PLA aggregated antigens? It's about 2/3. And they were already saturated. There was the same amount in the stage 4. That's the early stage here. So this simply shows that the PLA aggregates, the non-inclusion, they precede Lewy bodies and they affect way more of the tissue. And then I guess one should ask, okay, how does that affect the circuitry in this tissue? And how would that, turn into symptoms for the patients? And again, is this relevant for disease modeling? We have to ask ourselves. Also, if you see, let's say, for the PLA signal, there's quite a lot of the dots that are away from the cell bodies of the neurons. And then we ask, could it be that these aggregates are actually located to nerve terminals? And then Nana worked with Silvio Rizzoli in Göttingen. He has made this 10x expansion microscopy. And then we super resolved the signal. And then what you can see here is a pre-synapse that is labeled in blue with the PLA, so that's the aggregates. The red signal, that's a synuclein nanobody. So we have synuclein there where we have our PLA signal. And the V-glut simply shows that here we have a pre-synapse. So I think this is interesting to consider, could one have Parkinson's disease in nerve terminals and axons without having the cell bodies affected? And how would that affect the functionality of the cell? Then let's talk about disease modeling. I'm just following the time here. So often when we model synuclein pathology in our disease models, and that's also where we try to do our, let's say, our drug screens and disease interventions. The way many labs work now is that you actually add preformed synuclein fibrils to your models. And that can either be, native neurons, or it can be transgenic neurons or transgenic cells. And then what you score would typically be, aggregates of synuclein, for example, detected with an antibody to phosphosynuclein 7129, as depicted in the top right with the green stains, these beautiful dots there. So these are aggregates that are made within the cells upon addition of fibrils to the cells. And the way we believe it works is that these fibrils gets into the cells, they meet the normal synuclein, then they basically template and make it aggregate on top of them. And then you kind of catalyze an aggregation process. And then, of course, we hope this would mirror the aggregation process in the brain, and we can study the mechanisms. You can also just stain for, as shown in the lower right panel, where it says B. This is a mouse, transgenic mice, where you simply just stain with an antibody to synuclein and can actually achieve, Lewy body-like structures as well. But it's just to say that here we appreciate inclusions like we see in human brain, and we don't address this about could there be something that is not organized in inclusion. And could this be relevant? I think this is super relevant for drug screening, because we have in our lab, and there's probably a few others that has, but people don't often pay so much attention to it. But we have models where we have seen dysfunctions. For example, to the left, this is an SHSY5 cell that when you overexpress alpha-synuclein and it's in a non-mitotic state, after some days we saw there was a decrease in salicylic calcium. If we added an aggregate inhibitor to the cell, we could basically attenuate that decrease. So that was what gave us some confidence that what this effect was due to synuclein aggregation. To the right, it's another cell model where the phenotype was a change of the microtubules. They're shown in green. And the red cells, they express a protein that makes synuclein aggregate. So here we could show that when we made synuclein aggregates by co-expressing them with this protein, they retracted the microtubules. If we added this aggregate inhibitor, we rescued the phenotype. So if you just score for inclusion, you'll say these cells are fine. They don't have any problems. But then recently, using the proximal ligation assay that Nanas here have developed, we have tested the same cells that we showed where there was this calcium dysfunction. And here in this little busy figure here, we can actually now detect aggregates that are PLA positive without having any inclusions. And just to take you through it, so to the left, we have the cells where they do not express synuclein. We have not turned it on. Then you see no signals. If we turn on synuclein, that's the middle pattern, then you get these red dots. That's the fluorescent signal for the PLA aggregates in this particular assay. And if we add the aggregate inhibitor, ASI1D, then we lower the amount of aggregates. It's quantified to the right. So this means this model might actually give us, we might use this model in, let's say, drug discovery. But then you need, you can't score it for inclusions because you won't see them. So here, I think we should be a little cautious. Because, for example, if we're now trying to say, okay, what's the impact of synuclein aggregates in a cell? It could be, metabolic changes. It could be proteomic changes, transcriptional changes, epigenetic changes. Or it could be, if it was neurons, it could be changes in neurotransmission. That's the way, the brain is working. If these changes are associated to, let's say, the non-inclusion aggregates, then we might miss these correlations if we just want to score inclusions. Then, again, yes. Then we can say, can we appreciate synuclein aggregates from brain in biofluids? And what has been done in many labs for many years has been attempts to measure, and actually, people have measured total synuclein, phosphorylated and labeled or modified synuclein, or even oligomeric forms of synuclein in CSF, plasma, serum. And of course, the idea was, could we get a biomarker that could kind of correlate to disease progression, and we could study if we could affect that if we'd made some disease interventions. Let's say the result of all these has been that the biomarkers are not very strong. But then, of course, things develop, and then there's been this recent paper where they have studied not just what's in serum or plasma free, but they say, okay, let's try to look for neuron-derived extracellular vesicles. And what they did here in this paper that I think is a little interesting was that they were not studying Parkinson's disease patients, but they studied a prodromal Parkinson's disease state, that's this rapid eye movement sleep behavior disorder. These patients that has this disorder, a large fraction of them actually develop Parkinson later on. So this is a prodromal cases. That might be some that would be easy to work with if we should try to do disease modification, because they haven't developed the Parkinson's disease yet. And what you can see here is that actually the sleep behavior disorder patients to the right, they have a higher level of synuclein in their vesicles, in serum, compared to the healthy controls. So this is, I think, very exciting. Then there's another assay where we don't ask for the antigen, but we basically ask for an activity of an aggregate. It's an assay that is developed in the prion field. Nowadays, we call it, as it's said in the lower right corner, it's a seed amplification assay, SAA. It has also been called RT-QuIC or PMCA. Doesn't really matter. It's the same, more or less. But it's an ultra-sensitive tool to detect synuclein seeds, and seeds are probably aggregates. We don't really know, but probably. And the principle is simple. You have, let's say, a tube with monomeric recombinant synuclein. And then you add samples. It can be from brain, it can be from a biofluid, to your monomeric synuclein. And then you say, does this biofluid you add, does it accelerate the aggregation of these monomers you have in your tube? If they accelerate that a lot, you say, okay, we have pathological seeds in this sample. And that's, there's not more to it except that it's difficult to make it work well. But for example, here is a paper from 2020 that was, I think, pretty remarkable, because here they took cerebrospinal fluid from either MSA patients or Parkinson's disease patients or healthy controls. Then they applied them to this simple assay, added some CSF to the monomeric synuclein, and then they read the fluorescence trace using thioflavin. And what they showed was that, yes, the healthy controls were not aggregating. The Parkinson's disease patients aggregate a little slow, but came up very high. The multiple system atrophy or MSA cases came up early or fast, but did not reach that high. And what it showed was that, yes, they could actually discriminate Parkinson's disease from MSA based on the fluorescence, because it was the same amount of aggregates that were formed in the tube, but the MSA strain emits less fluorescence when it binds thioflavin compared to the PD strain. So here you could actually use this tool to differentiate between patients. And this is just kind of a more or less a summary slide for this particular assay. I mean, there's been a lot of very influential papers, and it shows that this technique has a huge capacity. The potential is huge. It has this very, very big sensitivity. It can be used to detect prodromal cases, for example, the REM sleep behavior disorder that I showed you before with the extracellular vesicles. It can also be used on patients with hyposmia to see, is it due to say, an early Parkinson's disease? And also, for example, if you have genetic carriers of LAC2 or GBA genes that are not yet manifesting the disease, it can also detect some of them. It also has this, what I would say from a more basic science perspective, interesting ability. You can actually maybe copy-paste the aggregates from the patient sample into your tube and amplify it and make more material that you can use for your studies. Of course, we need to be careful to try to validate, is it really the true structure or is it changed? There are data that suggest that these re-amplified structures are different, but they may carry some of the, let's say, the information that was present in the seed. The limitation is that the readout is kind of binary. Either you get positive or not. And we don't really know the structure of the seed, even though I told you this about the potential to copy-paste. And then I think what is very important is that despite being, so extremely simple, principally, it's actually difficult to run reproducibly. So you have to be a little cautious when you see these papers and try to see whether all the controls are in place. This novel staging system has contributed a lot, or this tool has contributed a lot to this newer staging systems for Parkinson's disease that has just been kind of published the recent years, that have been discussed for some time. Let's see here. It comes here. So these are just published this year. And I guess here they say, more or less, the premise is that Parkinson's disease and dementia with Lewy bodies are kind of the same underlying molecular neuropathology. It also says that they are both neuronal alpha-synuclein diseases. And if you kind of buy into that, then one could say you could actually have a stage 0 case. And that would be if you were so unlucky that you have a fully penetrant synuclein mutation that we know will give you an autosomal dominant disease. Then you'll be stage 0, and you could maybe be stage 0 for almost all your life. Maybe you don't, I mean, the penetrance is high, but you could in principle go through your life without getting symptoms. Then there'll be a stage, what you call stage 1AB. That's when you get biomarkers. And that's, for example, this seed amplification, as I showed you on the previous slides. So if we take out cerebrospinal fluid, maybe serum from these patients, and we can say, okay, now you get positive, then we know that the synuclein aggregation is ongoing. And then we might actually say, okay, now we need to do some disease modification. Or you could say, if you on the DAT scan can see that the dopamine system is dysfunctional, but you're still not had any symptoms, that will also be a stage 1. And then it progressed to stage 2 when you get your clinical signs and you get your classical diagnosis. And then, of course, it gets further on, and then you get functionally impaired. So now I'm getting to the last slide. So let's say this widespread non-inclusion synuclein aggregates, they call for attention to dysfunctions from the regions of the brain that we did not before consider affected. So for example, we said in Parkinson's disease, cerebellum is not affected, occipital cortex is not affected. Now we know that in some cases you actually have synuclein aggregates if we believe that the PLA assay is working. Then we also need to pay attention to dysfunctions in this region of the brain. And then we can say, okay, how can these dysfunctions arise? Well, I mean, this is not because the cells are lost, but you have these aggregates that are residing in nerve terminals. They're residing in axons. Maybe they haven't even reached the cell body yet. And there they may affect, neurotransmission and affect calcium signaling. This is one of the focus areas of my group. But it's just to say that you can actually have small functional defects in neurons that contribute to, let's say, the computing, but the cells are still there, and it would contribute or add to the burden of the patient. So this is, I think, what we should pay more attention to in the near future. Then there's this cautionary note. I think, if we just focus on inclusions in our models, we might miss important information, and that could, hamper our ability to do, important drug screens. So we may actually discard effective drug candidates because we say, okay, they don't work because, we don't change the inclusions. But if they just wipe out all the let's say, the non-inclusion aggregates, that'll be fantastic. But we need to look for it. And finally, the optimistic note here in the end, and that is that I would argue that, the non-inclusion pathology exists in regions of the brain where there's often no apparent neuron loss, and there's not really any apparent neuroinflammation. And that's why we haven't really observed them before. So these cells are viable. And that means that we may actually be able to let's say, reinvigorate them if we could increase their proteostatic capacity or maybe their tropic support. Or more simple, if we can't do that, maybe some of the symptoms that they give rise to are due to, let's say, disturbed ion fluxes that we may be able to treat with some of the drugs we already have at hand, but we just need to test them here. So I think this is really a reason for optimism to me. I hope you can share that as well. I think that's my last slide, so I would like to take questions if you have any. Should I lose my control? I'm in control.
Yes, I believe you still should be in control.
Yes, should I?
And we will go ahead and move on now to the Q&A portion. Thank you, Professor Poul, for your very informative presentation. We will now start the Q&A portion of the webinar. If you happen to have a question that you would like to ask, please do so now. Just submit a question in the Ask a Question box located at the bottom of your screen. Let's go ahead and get started. All right, our first question here is, are there any non-Lewy body aggregates of alpha-synuclein that challenge our understanding of PD progression and its impact on neuronal tissue?
If there are any non-new, well, I would argue that these two papers that was published by the Japanese group in collaboration with Danish Center, they challenge our view because they simply say that there's more of the brain that's affected. And I think that means that when we kind of say, okay, the patient gives us some symptoms, and they may be due to the areas of the brain that's affected, and we think that's very limited parts. But now we know it's a much bigger part. That would challenge our interpretation of, let's say, cause and effect, at least mine.
All right. Thank you very much. Our next question here is, would there be any association between Parkinson's and ADD or ADHA dopamine-wise?
ADD and ADH, dopamine-wise. I don't think I can answer that question because, I mean, I'm really not a specialist in dopamine function and you know, how that's working. So, I mean, you should talk to a better person for that.
Okay, sounds good. We'll go ahead and move on to the next here. How early do you think the brain get invaded if one considers spreading from the gut?
Yeah. There was data from my collaborator here in Austria, Bohammer, some years back, his group, where they had patients that had, a colon sample taken. And then they stained that. I mean, these patients later on developed Parkinson. They could show that some of these patients had synuclein pathology in their gut 20 years before they got the diagnosis. Now, then you could say, okay, at that time, 20 years before they got the diagnosis, maybe there was already at that stage non-inclusion pathology, let's say in the lower brainstem, that could have contributed to, let's say, sleep dysfunction and so on, depression. But that was not studied. But basically, what we showed with the early and late Parkinson's disease was that, years before you think here's something going on based on the typical spreading pattern, there's actually inclusions. But this is simply not studied yet. I think it should be studied a lot.
All right, thank you. We have two more questions here. The next is, why are these presumed low abundant but widespread alpha-synuclein aggregates important when they aren't detectable by immunohistochemistry?
Yeah. How can I say that? Let's, if you walk on a street and there are some big rocks, if you walk into them, you hit yourself. That could be a Lewy body. But if there's a lot of small gravel there and you have bare feet, then you'll actually walk pretty, uncomfortable. And that would compromise your normal way of behaving. So I think just the presence of something abnormal that we can show in our models has an ability to, activate certain abnormal process, for example, calcium dyshomeostasis. It has at least the potential to do harm. Of course, now we need to test, but how significant is that harm? But we know that the Parkinson's is actually, I mentioned at the beginning let's say these non-motor symptoms that are kind of a box of a lot of discomfort for the patients. And it's also evident from clinical studies that it actually detracts more from the quality of life than actually the real motor symptoms. So I think we should really pay attention to this.
All right, wonderful. Thank you. Okay, it looks like we have time here for one more question. What do we know about the structure of the non-inclusion aggregates?
Yeah, that's a good question. I think what we know, the non-inclusion, we know that it's more than 2 synucleins because you could detect it with this PLA that just had a synuclein antibody. We also know that they has an abnormal structure because we detect them very well with this antibody that we use in our PLA. But with that said, then I don't think we know much more. We are working on and together with others to make some gel filtration fractionation and say, okay, how big can they be? But this is really, this is not solid yet. So I don't think really we know more. They could be, are they associated to metabolites, vesicles? I don't know.
Perfect. All right. Thank you very much.
You're welcome.
Well, thank you again, Professor Poul. Do you happen to have any final comments for our audience today?
Yeah, I would say it's a very exciting time to study Parkinson's disease because, it's just as if it's unfolding in many directions. It can start here and there and everywhere. You affect systems in our brain we did not expect before. that creates a lot of hope for, disease modification and a lot of surprises for us. And then I would say the whole biomarker field is really also exploding right now. So it's a good time to do PD research.
Wonderful. Well, thank you again, Professor Paul Henning Jensen, for your time today and for your very important research. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until March 6th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.